All right. Hey, all, and welcome to our webinar, The Secret Life of Seals, Monitoring New England's Complex Coastal Inhabitants. My name is Brian, and I'll be your moderator for tonight's webinar. And I'm very excited to be joined by Rob Digiovanni from Atlantic Marine Conservation Society. He's the founder and chief scientist over there. Thanks for joining us, Rob.
Thank you very much for having me.
Definitely. And we have Ashley Stokes as well, a familiar face, our marine mammal rescue manager here at the Seacoast Science Center. Thanks for taking the time tonight, Ashley, as well.
Always, anytime.
Yeah, sweet. So tonight we have some really interesting topics we're going to be talking about. So for context, back earlier in the winter, the Seacoast Science Center, our marine mammal rescue team, actually was able to put a satellite tag on our first harbor seal. This was the first seal that we've ever been able to satellite tag out here in New Hampshire that was rescued in New Hampshire. It was released up in Portland, Maine, thanks to after some rehabilitation from our friends at Marine Mammals of Maine. And in that time, it traveled from Portland past Boston, around the Cape. It was spending time in Nantucket for a while, and now it's back in Portland. We're going to show you all about this. But really what we're going to speak about tonight is the context of this research. You know, why did we decide to put a satellite tag on this animal and try and figure out where it's going in the Atlantic Ocean and the Gulf of Maine? So that's really why we're here tonight, and that's why we brought on Rob, who is an expert on this. You've been doing research for how long have you been doing this research?
I say greater than 25 years now. It just sounds a lot better. I know. Dinosaurs first, then me. I got it. Thanks.
So you have a lot of experience. And yeah, so Rob, if I figure we can maybe start with just how you started this research, why you started doing some of the seal tagging. What were you looking for 30 years ago or 25 plus years ago?
Well, actually, it's a really good question. And when you think about working in rehabilitation like you guys are, as far as responding to these animals, and you look at all of the questions you have and then figure out, okay, we put all this effort in, where do these animals go? Well, 25 years ago, there wasn't a lot that was known about what you do during rehab or after rehab, and do these animals actually survive once we put them out? I mean, it's great, and we all like to go and see a release and see an animal go, but we don't really hear a lot about what goes on after that. So we started exploring, okay, how do we find out what these animals are doing? And so we put flipper tags on, which are some tags that you guys have seen, but you don't really get a lot of returns. You usually get less than 5% return, so it doesn't tell you a lot about that. So there were other ways that we started to look at this, and for me, the idea was, okay, we have technology where we can put radio tags on, you can put satellite tags on, but it's all very costly. And so one of the things that we started with was just saying, okay, we don't get a lot of information from the flipper tags, and we really need you, the public, to help us by reporting what you see. So that's where we took our first step and started to use these things. They were developed over on the other side of the Atlantic by Hall et al. In 2000. They're called the hat tags, and I think we might have a picture of one of them coming up. And so you can see these hat tags. And basically, it looks like a little cheese head if you happen to be a Green Bay fan. Basically, what we did is we used these to epoxy them to the top of the seal's head, right onto the fur. And then what was interesting is more people would see these and think, oh, someone's doing something weird with seals, and they would report it. So we started to get a lot more information about where some of these animals go. And so that made us a little bit more comfortable with the idea that, okay, maybe we can take these seal hats cost a couple of bucks to put on. Actually, a lot of our colleagues have actually moved from the plastic hats to making them more out of wood, making them a little bit lighter. But then we felt a little bit better about two things. One is that they are seen a lot better. And here's a great picture that was taken by a colleague of ours off of Duck Island. And this happens to be a gray seal that we satellite tagged. And you can see that I'm sorry, not a satellite tag, but it has a hat tag on it. And you can see the yellow hat tag pretty clearly underwater. But what's odd about this, and this is where the luck or my luck really goes, is on that hat, it actually has numbers on 2 sides and who to call if you find it on a 3rd side. And I guess we can pretty much figure out which side is actually facing the camera. It's the one that tells us who to call, and we already knew that it was our tag. So we don't know which animal this was, but it kind of led us to more of our understanding that these animals that are being released down in New York are moving back up north, but we don't know how they're getting there. So then that's when we move towards more of the satellite tagging. And a satellite tag runs anywhere, between, for a very basic one, from $2,000 to almost $5,000. And then you have satellite time. You attach them to the fur. The thing with the satellite tag is you put it onto the fur with a 2-part epoxy, and then it'll stay on until the animal molts. But it gives you an understanding of where the animals go, not just point A and point B, but how they move through that area. And so our question is a very long answer to say our real question was looking at what harp seals do once we release them here in New York. And the argument is that they don't really do well. You can't really release them in New York after May or June because they don't go back up. And what we found is not only can you do that, but they go back up into northern Canada, way up into the Davis Strait off the west coast of Greenland. And they usually do that within a week and a half or 2 weeks. So they're getting way back up to where we would normally expect them to go. So once you go through the rehab process where the question was, well, once you deal with these animals, they don't function normally. Well, we don't always know what normal is, but we do know that they know how to get out of Dodge and got back up to Canada where they kind of came from. So they seem to travel great distances and they navigate at sea level, something that we would have a hard time finding those places in Canada.
Yeah, that's really interesting. I know something that really stuck out to me in some of our conversations before this was the need to have, you've been doing this for such a long time, the need to run this study and this research over a time span of more than 10 years, more than 20 years. I found that really interesting.
Well, yeah, because the biggest problem you face with anything that you do in science is basically, is this an anomaly? I mean, let's go back to the seal hats. We put out, what's funny is we put out, I think the first time we only put out like 8 of them, and we got calls on 3, which is a really great return. But you look at the second one we got, it didn't have the number on it, so it didn't tell us a lot of information other than a seal that we tagged went all the way back up off of Maine. So in that case, you have a lot of variability associated with this. So the only way you can start getting some more confidence in the answers that you're receiving is by increasing your data set. And it takes, just like it takes anything, a time to rehabilitate an animal. Animals come in for different reasons. What are they doing? How are they here? How long at the time of year? So there's a lot of variability associated with that. And what's really exciting about what we're able to do now, especially with Seacoast Science Center and even with Marine Mammals of Maine, is that by working together on a lot of this, we can build these data sets and start now applying a lot of these data, not just to the rehabbed animals, but to some of the wild studies that we're doing for health assessment to look at movements, which can answer a lot more questions than, or I should say, about 4 years ago, I actually sent an email to a colleague of mine that I've been working with for greater than 25 years. And there was like 5 questions. And I just said, are we still trying to answer these questions? And they were pretty basic. And her one line was, yep. I was like, okay, just making sure that we didn't miss something. And it's, it's a big ocean out there. These animals move great distances, probably a lot more than what we give them credit for. And all of this work shows that. It shows that we have an animal that we released, went all the way down south, and came all the way back north.
Yeah, absolutely. No, and we're going to come back to some of those points that you were hitting on in just a second as well. But next, I want to move over to Ashley here real quick, and kind of discuss how teamwork does make the dream work. How our rescue program, like, where do we fit in with this research? Why are we so important to this equation?
Yeah, that's a great question. So if you think of all of these individual organizations that are doing this work, if everybody just does their piece of the work, now our network runs from Maine through Virginia. So if all of the organizations within that network and even outside of the network just did their own research and their own projects, they're only getting a small subsample of these populations of animals. And if they're not sharing that data and collaborating on that data with other network partners, what are we really learning? Whereas if we, teamwork makes the dream work. If we branch out and we work with our colleagues within the network and we work on questions, solving questions together and expanding those data sets, if we're only looking at animals that we pick up in New Hampshire or Northern Mass where we respond, that's fine, but we can do a whole lot better if we're looking at animals that are picked up everywhere. And different species, do these animals, once they're released from rehab, I've always been curious, do they act like a wild animal? Do they travel as far as a wild animal would, or do they stay around one spot if they're not comfortable? So this has been really important for our program, but it took a little while to kind of get started. So our program started January of 2014 for marine mammal response and rescue, just in New Hampshire. And then in 2018, we expanded into Northern Massachusetts, doing the same work as well. But then this year, we were fortunate enough to be able to start to add, and really work with our colleagues and different partners, the satellite tagging aspect. It's something we've never done before. I don't know how many animals prior to our program starting had been tagged, but it's something that we were really excited to get into. Of course, Rob mentioned the costs of the tags and the satellite time and everything. Rob's, travel time for different organizations to travel and do this work. So it certainly all adds up, but it's something that's really important. There's so little that is known for such a very forefront animal species. I mean, everybody knows what seals look like and roughly how they act. And, we all hear about the seals down on the Cape. So it's important to start unlocking these doors and learning more about these animals that we only see a small amount of them that have to be picked up and worked with. And, now that Rob's doing wild caught animals and tagging those, and we can, really start to broaden what we're learning and start to understand about these important animals.
Yeah, that's a really good point, Ashley. And just some of that video that was in the background, really just showing, our rescue team, and that's really what we do on a daily basis out here, is work with those animals.
Right.
And this, like you said, this unlocks new doors for us to be able to contribute to greater scientific research with our friends down in New York. And don't worry, Rob, we still like you, even though you're from New York. But so with that, I kind of want to move on and ask Rob a little bit more about kind of what were some of the goals that you have in collecting this data. So we touched on this a little bit, talking about the difference between how do rehabilitated seals behave post-release compared to wild caught, not wild caught seals, but wild seals, right? And that was one of those big questions that you have. And I'm curious if you could speak a little bit more to that as well.
Sure. I just actually, one, I think it was a great video that you had looking at the tubing of the seal. And one of the things that we've looked at, and I was kind of chuckling when you talked about teamwork makes the dream work. We were saying teamwork is what makes our network. And when you were showing the dose syringe that you were using, the big syringe, we started using them many years ago because that's what they used to actually rehabilitate dolphins at National Aquarium in Baltimore. And so I went down there one day and I was visiting our colleagues down there. And I was like, hey, that's an awesome idea. We're going to steal that. And because we didn't. And so that's how we started doing that. Because before that, you used to pour it into a tube out of like a blender. And so after wearing so many shakes, you're like, there's got to be a better way. But it took our whole network coming together to say, hey, this is how we do this. These are the lessons learned. And that's really what I think comes back to. We all kind of learn how to rehabilitate these animals, how to respond to them, what works, what doesn't. But then the biggest data set that we had for the longest amount of time, because we decided to start tagging, was actually from stranded animals. So the very first, it wasn't one of the first animals. The first animal I satellite tagged was actually a hooded seal. And so that they just kind of do odd things. But we had a gray seal many, many years ago that was freeze branded on Sable Island and then stranded in New York. And I was working with a colleague of ours, excuse me, Greg Early. He was at New England Aquarium at the time. And he had I said, do you have a satellite tag? We know where the animal came from. Why don't we see where it's going to go? So we rehabilitated the animal. We put the tag on, and I know New York, nobody, great you like us there, but so it's way down south, way far away from Sable Island. And that animal goes off the beach and just heads due south. And so this is kind of around, email kind of thing, but we're old school. So we were on the phone every day. Our call was, oh, this isn't good. Oh, it's not doing what it's supposed to. Oh, this is really bad. And then it gets all the way out to the shelf break. And then it just makes a left-hand turn and drives and just swims all the way back up north to Nova Scotia. And like clockwork, we go, or that's what they do. And so that's how little we knew about what these animals would do. But it's also showed me that we don't give them enough credit for, yes, they come in, they might have issues that they're trying to deal with, but it doesn't mean that they forget how to be a SEAL. They don't just all of a sudden say, oh, I don't know what I'm doing now. It might take a little bit of time. And to see this and see that this animal, I mean, this is a young animal, just born in the previous year, and it headed on back up to that area. So it gives you an idea that these animals move around. But now, from some of the work that we're just doing this year, even in the last couple of years, we've been tagging some gray seals as well as some harbor seals. But we satellite tagged a harbor seal in Shinnecock Bay on the south shore of Long Island. And the day after we tagged it, it left Long Island and went down to Delaware Bay and stayed down there basically the whole time until it decided to head all the way back up to Maine. So all of this behavior that you would see from what you would consider rehabbed animals and say, hey, that looks a little odd. Now we're seeing similar behavior in some of these animals. It's still early because, your sample sizes are small, but it's exciting because we finally have sample sizes that we can compare these animals to. And that's where all of this, we can start asking a lot more questions now because we're working at this as a team and being able to increase that effort and that sample size.
Yeah, that's a really good point. And just thinking of this now, I mean, do you have enough data in this data set to be able to come to any conclusions about these animals so far? I'm just curious. Like, do some of those? Does every harbor seal that you see that you tag travel, between state lines, or do some of them stay in one region?
Well, everybody's gonna say we need more data, but our sample size for wild animals is still what I would consider small. And I really smile only because there's a part of we went to the seal hat idea. There's a part that I left out that I originally thought was a great idea, was to use radio tags because they were a lot cheaper. And so what I did was I purchased a radio receiver. I put it on a tower in Shinnecock Bay. And then all the animals that we released, we put radio tags on. And the one thing I found out is they left Shinnecock Bay within a day and they didn't stay anywhere near the receiver. And that's where we realized that they move a great distance. The problem is my argument's always been that they move a lot more than what we think. So in a sense, I was right, but it still was like, okay, now we have to go and try using these satellite tags. What I've always found that was interesting is, and getting a little into how satellite tags work. I don't know if you can see this. I have a satellite tag here, but it kind of looks just like this here. And it's got an antenna. And when it comes to the surface, like if this is the surface, it comes to the surface, it'll send a signal up to a satellite. That's how you would get a position. And what was odd is we would always get a lot more positions around these haul-out sites that we knew seals were hanging out. And this haul-out site is where seals climb out to sun themselves, rest, or whatever they would do. And so we would get a lot of points there. So you would see that they would start moving up. So I think it's pretty fair to say that these animals use the Northwest Atlantic throughout their range. They move regularly. There might be a seasonality associated with how far some of them stay, but just like not everybody goes to Florida or North Carolina in the winter. Maybe there's different age cohorts. Those are the data points that we need to really tease out. I think the much broader understanding that they move up and down, and then there's a lot more mixing between areas, and even with some of the other health assessment studies that we've been part of, we found differences in different haul out sites. So, I mean, there's a lot of data that's really coming together. I don't want to sit here and sound like more, but the point is that we're able to ask those other questions as opposed to, do they just go back up to Maine? Now we can say, when they're going back up to Maine, where do they stop? Do they have issues with disease transmission? How does that work? So these are really helpful tools when we think about some of the things that we've encountered over the last bunch of years, such as the unusual mortality events and how does that spread. So yeah, I think that we're learning a lot, but there's still a lot more for us to learn.
Yeah, definitely. And with that, I want to transition over to diving into some of the maps that you've been able to generate from our animal number 87. And I know that our audience out there has been, following our updates that you've been sharing about its travels. And I'll pull up this map now. So I figure maybe Ashley, if you want to discuss a little bit about this animal's travels here. I know this map, some of the colors are, they go by month. But yeah, if you want to speak a little bit about where 87 has traveled and why that's interesting to us.
Yeah. So if you had asked me, so this was a young animal, number 87, that was picked up in Rye, New Hampshire. And if you had said, Ashley, where do you think it's going to go when we release it in Maine? I would have guessed, being such a young animal, that it would have stuck around for a while to get its bearings again, figure out good areas to forage, good areas to haul out, those kinds of simple things that these animals are doing on a daily basis. I would have never guessed that we would release it in Maine, and then it would very quickly move out of the area. So you can see, we released it in February, and it very quickly started moving south, which are indicative of the red dots. And then by March, it stayed down below south of Cape Cod. That right off the bat was a shocker for me. I would have guessed at least the first few weeks it would have stayed up in the Maine, maybe New Hampshire area, but I didn't think it would stray too far. So maybe that's just me being naive and, the data and being excited about our first satellite tagged animal. But this animal has shown me personally that these young animals are capable of so much. I mean, the miles that this animal has covered since being released to now is just amazing. And to think that it's back up now in Maine, it makes me wonder, where's it going to go next? Is it going to keep going north, or is it going to hang around the area, or is it going to turn around and go south again? We just don't know. So we'll have to wait and see.
Yeah. And Rob, I'm going to share this other map that you generated as well, which compares the path of number 87 with another harbor seal who was released at the same exact time, which was it was rehabilitated with Marine Mammals of Maine. But this one, in this map, the blue animal is number 87, and the red animal is this other harbor seal that was released at the same time. And they took very different paths. I don't know if you could speak to this one a little bit more. He spent some time closer to home to you.
Yeah, actually, this is really a trick that I played on all you people, because those are the colors for the Mets uniform. It's more blue and orange, but I thought it was funny when I was putting it together. Yeah, I think that what Ashley was saying about, I had the first thought when we were releasing animals that they wouldn't travel as far, but to see that the one animal comes all the way down and was hanging out south of Nantucket, and this other animal went and even to all the way west. What's interesting about that in western Long Island, the fact that it went through Long Island Sound, and then it's on this, hanging out in this one area by Sandy Hook, New Jersey, and this small island that just started to have seals start hauling out in the last decade. It's called Swingburne Island, and it's just south of the Verrazano Bridge. And it looks like this animal was taking up a lot of time there. And we always get concerned when we see the animal hanging out a lot, but then all of a sudden it just decided to start heading east, and now it's in the same area where south of Nantucket and starting to head back up north. So this is one of the reasons why we talk about needing a larger data set, because 10 years ago or 15 years ago, we would say that, oh, both of these animals did something that's odd. Now we're saying both of these animals went to places where we know seals are now. So they both actually did something different. But just like I'm using Florida as an example, just like some people go to Miami, some people go to West Palm Beach, some people go to the West Coast, these animals move and travel in different ways. But yet all of these dots that you see for the animals, you see them when they show up near shore, they're showing up where there are haul-out sites where seals are normally seen. So I think that that's what's very interesting, is that we're seeing them, that they find it. So think about this. I don't know where any of us have never gone, but just tell me where to go to eat in that town where you've never been. That's the interesting concept. These animals find that. And how do they do that? I don't know. I mean, many of the animals we've tracked recently, we've actually seen them go right through Cape Cod Canal. Now, if we were out boating, we can't find that canal without a chart, but they find it from sea level. So there's a lot more to what these animals are doing than what we're understanding. So once again, it comes back to, yes, if at a very basic level, we can look at seeing what they're doing, but we're learning more and more about how their behavior changes. And everyone, I mean, 20 years ago, you wouldn't be thinking of seals in Western Long Island Sound.
Yeah. Well, that's exactly what I was actually going to mention is, I mean, I don't know if I grew up in Long Island as well. And I know that this animal, it must have traveled through the East River essentially. Right. And I know if you said that to anyone who grew up, any amount of time ago near the East River before the Clean Water Act, they would tell you you're out of your mind, you know? So I don't know if that's, if you've seen, that they're spending time near shore in those in densely populated areas because the water's cleaning up, because the environment's changing. But I assume that these are all questions that are on our radar in terms of what we're thinking about.
Yeah, they're really good. They're really great questions, actually. And we can always look at this and say, well, things can always get better. We definitely have to improve our environment. We definitely have to clean up a lot of the mess. I mean, in really awkward terms, I mean, it's like my mom always said, no matter what, if you go to a party, you should always help clean up. And, it seems to me like we're not really doing that as well as we should. So we definitely can be cleaning a lot more. And there is improvement. But the other part is there's improvement, and there's also more food around. We're seeing a lot more menhaden or bunker. So we're seeing a lot more sources for food. And I've used this example not just for seals, but for whales as well. I mean, if you're older like I am, you kind of remember traveling up and down the interstate highway system when you went in and there was a vending machine and you got coffee out of a vending machine for, a dime or something like that. And it really wasn't Starbucks quality. Well, now you go to so you're not going to spend a lot of time there, right? You're not going to hang out in those areas. But now you go to a rest area and you can get a vegan meal, you can get just about anything. So you're going to spend more time in those areas. And that's probably a lot more like what our habitats looked like years ago. People didn't come here because there weren't any animals or there wasn't an abundance of wildlife. They came here because there was an abundance, but it's probably a heck of a lot more than what we have today. So it's interesting to see how these animals are moving back to those areas. And it's really a huge success story because you have a lot of negative impacts. Then you have a lot of conservation efforts that are going on, a lot of heroic efforts from rescue programs like yours. And then you start to see positive feedback where you see more animals in these areas, which is what we wanted to see as a result of those efforts. So, I mean, we're seeing some really great things that 30 years ago you were thinking, oh boy, we're really doomed. So,
yeah. So it's not all vending machine coffee anymore.
No. Unless you like that, because I'm not being judgmental on it,
Yeah. No, that's funny. But that transitions really well into the next thing I want to talk about, which is, we're collecting this data for a reason. Are there any conservation implications to this satellite tracking data? Is there anything that you're looking for? For example, do you forward this to NOAA? looking at fisheries overlap, for example, or development, any of that kind of stuff?
Yes, actually, that's where there's a need for partnerships. We all work and conduct a lot of our work under, in part by the stranding program, under our stranding agreements, but in part from the wild capture work under our scientific research permit. And we partner with the Northeast Fisheries Science Center and NOAA Fisheries on all of these projects to coordinate the effort so that we can, even to the point where we try to program our tags in a similar manner so that the question that I might have might be able to be answered by them programming their tags in a certain way like I do, but then they can answer a totally different question. So I can look at, okay, do we see these animals? Where are they hanging out? Not in relationship to food sources, and they might look at other issues such as where are they hanging out in relationship to food sources or bycatch or any of those factors. And that's only going to help us understand what these animals do by increasing those sample size. And I don't ever want to sound like I'm saying we just need to do more and more and more. But the biggest problem that you have is you might just be very fortunate to get the only animals that just hang out and don't go too far. And when we first started tagging gray seal pups, it was a similar conversation to that phone call that I had about, oh, they don't do that. We thought that those animals would kind of just stay around in the area and not go too far. And we found out that they went in and out of the water, in and out of the water, and then some went right up to Canada, some went down to Delaware Bay as well. So these animals do move around, but not all of them. So once again, as you look at the population and you try to understand how are they interacting in the ecosystem, you need to know that a very simple back of the envelope calculation, we tagged one seal in our first attempt in January, and that animal would never have been seen in that haul-out site again through any of the other works, whether it be aerial surveys or anything else, because it went to Delaware.
Yeah.
So that tells us that they move throughout this. So it actually supports some of the information about looking at these more as a basin-wide, what do the animals do, as opposed to just, do you have animals that are just hanging out in New Hampshire or in Massachusetts? And so we try to answer that question.
Yeah. And also to, I've seen to bolster your sample size and to compare to wild populations that you have been satellite tagging, wild animals that haven't been in need of rehab. I'm curious just how that process, how do you go about that? I've seen you use some kind of a net that you've designed? I know anytime an adult seal, when they're resting on a rock, as soon as they see you, approach, there's a reason we tell people to keep back, because you're going to flush them into the water. They're very reactive to people. So I'm curious how you go about that.
Well, I think, and taking a huge step back, the biggest part of this is that, from the partnership, I mean, how do we catch seals and how do we go out and do this? This would not have been possible had we not, I started off working with the Science Center. I think we did our first real capture work back around 99 or 2000, working with Gordon Waring at the Science Center. Now we work with Kimberly Murray there, but working with Linda from Marine Mammals of Maine. And I put these people out there is because it really just took this mindset that said, we're going to try and do this no matter how difficult. And I think you had to forget about how foolish you're going to look when you don't catch them, because they're really good at not being caught. So, how do you do that? We have a number of vessels that we use, small boats. We go out and we use a big net, and then we would encircle the animals. We would seine them up. We seine them up onto the shore, and this is a picture of us in the project that we were working on here in Shinnecock Bay. We're working on this pontoon boat with our colleagues at Stony Brook University. And so we were able to work the animal up on the boat and put a satellite tag on, and we were taking other samples such as cultures. So we took nasal cultures to look for bacterial or viral issues. We drew blood, and then we released this animal back out into the wild. So, it's the how is really, first is you have to decide what you want to do and then go do it. And then always make sure that what you're doing is really answering a valid question. And that's where we are right now, is that there's so much information out there that says, oh, these are the same seals that we saw yesterday. And I never really prescribed to that thought. But most of our data is supporting that they change through, they rotate through. I always believe that seals at Shinnecock Bay used to go and hang out in Mauritius Bay. We had very little data to prove that. Those are two bays that are close to one another. I put one satellite tag on, and based on the tag, it looked like it went right actually through the back bays to go to the other one, but it was one animal. That was very unusual. But then working with our partners down here, Coastal Research and Education Society of Long Island, Dr. Artie Kopelman, he's been looking at doing photo ID of animals for many years. And I mean, he loves the story because I actually did try to catch a seal, put a tag on, and it didn't work, the tag. It just didn't start up. But he saw that animal for 10 years since we tried that many, many years ago. So we know that the animal came back. So, it's part of having other groups working, citizen scientists telling us what they're seeing. I mean, this cannot be done alone, and everybody is a crucial part of these projects.
Yeah, definitely. And so just also, I know that people probably have this question about the satellite tagging. Our animal, for example, is still out there transmitting that tag. When will that fall off? And I don't know if you could maybe speak a little bit to how they're applied. I have some video footage from when we applied this tag with you that I can pull up here. But I figured it'd be great if you could speak a little bit to that, about how much longer we might expect for this number 87 to keep transmitting.
So that's a really good question, and I kind of make a face because I always think it's, you always want it to stay on for 30 days, and you want it to stay on for 60 days, and you want it to stay on. Because you can see we're using a two-part epoxy, and we're putting the epoxy on the fur of the seal, and basically we kind of mix it into the fur, and we put the tag on, and then this will harden and it'll stay on until the animal molts again, or it just comes off. And it's kind of, think about it as like getting paint in your hair. Eventually it'll come out. So I've seen animals that have basically had a tag put on and they rolled around for a little bit on the tag, but they tend to get used to it pretty quickly. I don't think that it really has a huge impact on them. One of the things you might notice here is that you can see that this animal obviously has a tag on it, a satellite tag, but it also had a flipper tag. So the flipper tag will stay on a lot longer, whereas the satellite tag, we usually, getting a year out of a tag would be awesome because it's attached to the fur. But when we get anywhere around 90 days, I think that we have a pretty good sample size and a duration. So these guys are doing really well.
Yeah. So number 87 is still hanging on out there.
Yep. That's great. I mean, you could think about maybe towards midsummer, you might start to lose it. I wouldn't be surprised if we did.
Yeah.
But once again, those are some of the questions that we're also asking, because we have a lot of information about when they lose them from rehabbed animals. Is it different when we're putting it onto some of the wild animals? And is it different, just like you have animals that might molt at a little different time if they're at higher latitudes, so if they're higher. So there's a lot of that variability associated with this that we're learning a lot each time. So, I mean, I think it's great. And I really do. I thank you for letting me be part of this project to go do it. It's, great. Because even though I'm from New York, I like coming up north too.
We like having you.
Oh, definitely. And with that, I have a few closing thoughts. But actually, I wanted to ask you, what's the future for our program here at the Science Center? I know we were able to, number 87 was a perfect candidate for us. Right. And we were able to get some funding from NOAA, from our Prescott grant to really help make this happen. Are there plans for future tagged animals at the Seacoast Science Center, if they're good candidates, and what makes a good candidate?
Yeah, that's a great question. And it's multifold, I think. So yeah, some of this funding did come from a grant that we applied for, federal grant. But, Rob mentioned how costly these tags are. So luckily that grant has covered the satellite time. So it does take money. It's one thing to put the tag on the animal, but it does take Rob and his team time to get the satellite data, have it transmit, digest that data, make the maps, travel, up to us, all of those things. So it's not just the cost of the tag, that goes into a lot of this work. So I am hoping that we will be able to tag at least 1 or 2 animals a year moving forward, so that we can be contributory, to a lot of this research that's going on. But it does come down to the funding. So we're hopeful that, we have some partners out there and maybe some donors out there that would be interested in helping us continue this work, as we, continue to run our program and find these candidates. You have to be mindful of, as Rob mentioned, when these animals molt, and, is the tag going to fall off as soon as we put it on, and who the right candidates are. Are they animals that are good to be carrying these tags, and things like that. So it's very exciting that we've done our first one, but I'll be even more excited when we can say, we've done a dozen.
Yeah. Oh, agreed. I think it's been so interesting. And even in terms of education here at the Science Center, I know just being able to share these, that's another component to this. It's not just research, it's also education. Being able to share and get people to actually see where these animals are moving. It makes you a believer, it makes you see these animals in a different light. But yeah, I agree. It's very exciting stuff at the Science Center. And Rob, another one of my closing questions for you is, what does the future of this research look like? How do you see it, going 10 years down the road? There's a lot of, I feel like people have more questions now than they really ever had about these, iconic coastal animals. What do you have, or what do you see?
So that's a really good question, because I'll still be answering it. I've been doing this for 25 plus years. So 10 years from now, I'll still be giving you the same answer in that one. But as Esther's talking about a lot of the work that goes into it, I don't know how many of you have worked in DOS or programmed in DOS, and the first tags were programmed in DOS. So it was all zeros and ones. And if you missed 1, 0, or 1, you either turned the tag on or turned it off, or it didn't work. So, well, that took a lot of time, and nobody cared. Nobody cared about it until you deployed the tag, and then everybody showed up at my door saying, "Where's the animal? Where's the animal?" Well, we also had to process a lot more processing after that to get these things. But the thing was, it showed me how interested people were when they could see where their animal went tomorrow or the next day, and then it engaged them in the community. And then we have discussions like we do now. Is our water quality getting better? Is there something we can do to make the environment a better place for these animals? And the answer is yes, but awareness is a big part of it. So I would never be able to really say which is more valuable to the ecosystem, is the educational awareness of how these animals interact with it, or the data that we get about how they use the habitat. Where do I see it going? I think if we were kind of looking in a crystal ball, I mean, the future is boundless, because I just explained we started what we're doing right now with DOS, which would take days to get you, not a map, it was kind of a sketch, that we would kind of come up with. And now we can do this a heck of a lot because of a lot of the programs that we have. We have Critter Cam that's out there to be able to look and see what animals are doing below the surface. And so, we also have, the GoPro cameras that show us so there's a lot of things that we can bring together. And this is where I feel, when I'm always asked about how do you get into this field and, what do you do? I mean, the diversity of individuals that we need to work in this field, and the diversity of talents that we need to try and explore the technology and how do we apply it to answering these is boundless. And I think that's really where the future comes in, is that, whether it be a programmer that knows how to work with these data sets and bring in satellite imagery data, or it becomes someone who does media or video and says, hey, we want to do this. I mean, I'm from the generation where people say, how did you get involved in this? And we all remember Jacques Cousteau, which was more of a cinematographer, and telling us what was going on and having us expose these wonders. And that's where I see these animals are definitely sentinels for what's going on in our environment. And we need to really start listening to them a lot more and trying to understand what's going to happen to them is happening to us in many places. So the future is on us hearing what the environment is telling us and trying to learn as much as we can.
Yeah, I think that's a perfect answer. I mean, and I can't think of a better way to kind of close up this webinar about the secret life of seals. I mean, you hit the nail on the head. There's the technology's changing. There's so many ways today that people can get involved and support both the work of both of our organizations. So I would say if you're out there and you want to step up and help support this work, whether it's the rescue side of things, you can reach out to us and Rob at AMCS, and getting involved with the research that Rob does down in Long Island. If you're from Long Island and you see this, definitely reach out, and we can always use the help of anyone out there, no matter what skills you have, just like what Rob said. So I don't know if there's anything else you want to close with. It's going to be tough to beat that one, but
I thought you saying don't say anything, Rob. Be quiet.
Yeah.
And for anyone watching live, if you have any last minute questions you want to sneak in, just feel free to comment. It will pop up for us, and we're happy to answer those.
Yeah, definitely. And thanks, Rob, for taking the time. It's always a pleasure to have you on here and to gain some insights from your infinite knowledge on seals.
Well, it's great working with you guys. I really appreciate it. I think this has been a great project, and I'm really excited for where it's going and for you guys taking these steps to collect these data. I mean,
yeah, definitely.
That's how we all started.
Yeah, follow along. We'll have more. Hopefully, we have some more animals, out there that are being tagged. So we'll keep the data set alive, and we'll keep that number growing. Rob, we'll see what we can do for you.
There you go.
Perfect. So thanks for tuning in, everyone.
Thanks, everyone.
Thank you, everybody. Have a good night.